And I wanted to talk to you about some work, more recent work, that was headed off by Clay Kressler, who was a PhD student of Aaron King, my collaborator, and he did the mother of all simulation studies. So I wanted to tell you a little bit about that. We learned some interesting things about using the OU model to detect adaptive evolution. So this is specific to comparative data. And we learned a lot about model selection and parameter estimation. Okay. So there's been rapid development of OU models in comparative analyses. So of course, you know, Ornstein and Uhlenbeck developed it in like 1930, and then it was used by quantitative geneticists, Russ Landy's papers in the 70s, something like that. And then Hansen in the 90s first applied it to comparative hypotheses. So we added on to that. And then we added on, we really kind of fleshed out this multiple comparison thing, multiple model OUs. And then also Hansen also continued to work on this and developed a different system called Slouch. And but it's sort of a more, it's the same thing really, but it's in a more regression framework. And so he thinks of it as a hybrid BM-OU model so that you have a evolutionary regression between a phenotype and the covariates. So we like to use the painting of the optimas on the phylogeny. He likes to express the same ideas in a regression model. So it's just like whichever one makes more sense to you, fine. You know, they're basically doing kind of the same thing. Both groups have developed multivariate extensions to these things, and then AUI, which is Brian's package. So introduce. I'm sorry. Okay. He's being very modest, but anyway, AUI introduces the ability to have both multiple optimum as well as varying alpha and sigma across the tree. So you can see everybody wants to complexify, everybody, right? We're like, we got this new toy, let's see what it could do. Okay, so so here I want to make some comments about the different ways that people use these models for different goals. So our group, our collaborative group, we always are thinking about the evaluation of biologically informed hypotheses. We have some notion about the life history of our organism, and we want to test these ideas. What explains muscle fiber type? Is it foraging mode or is it predator escape mode? What affects body size? Is it the habitat type or is it the diet? You know, things like that. So in this case, model selection at the end where you're comparing all your hypotheses against one another, it's an aid to reasoning. So it's our basically critical thinking about the importance of specific evolutionary mechanisms and drivers. So it's a tool, not an end in itself, but it helps us to see, do we have any power to support one explanation or another? You may or you may not. So it relies very strongly on the construction of a very small number of well-reasoned and hopefully distinct hypotheses. Of course, if your hypotheses are almost identical, you will have a very hard time distinguishing amongst them, right? And then the larger the number of models you consider, the less reliable are the inferences that you get. Okay, you just can't run statistical tests one after another and expect that you're not going to degrade your power. I mean, it's, you know, if you test everything, something will fit, right? Okay, so this follows along, you know, Burnham and Anderson's work, and all of these people have commented on these things. other people are not really so concerned about testing biologically informed hypotheses, but instead they want to basically find out the best reconstruction of ancestral states. Or they want to see, like, where have there been changes in the history of the taxa. So this is a different goal. And so they want to infer the ancestral states, or you can think of it as historical shifts in selective regimes based on the phylogeny and the traits. So in this frame of mind, you want to avoid the a priori limitation of the range of possible histories. You don't want to make hypotheses. You want to see what's there. And that makes use of an exploration of a huge number of alternative histories to find those most consistent with the data. And this is, I guess, how I would characterize Bayou and other, so I need to add that. But other people have been also thinking about along these lines, and there are various software implementations of this that explores shifts to find where they are. So both of these are valid, laudable goals. However, the one danger is what? Yeah.
That it may be fit statistically, but it's not biologically feasible or that it's not sensible when you evaluate.
Okay, yeah. So where would you violate the statistics, though? How could you use these in combination where you would violate statistical thinking? Yeah. And so what if you found the very best painting that fits your data the best, and then you test it against some alternatives and got a very significant result? And then you're like, so why is that wrong?
Testing it against very, very bad alternatives that aren't.
So why can't you find the very best fit first and then test it against some other alternatives and then be very happy with that, let's call it a p-value?
Well, you're kind of confirmatory at that point, I guess. If you found all the best fits and then you later find that it's still best fit, that's really surprising.
Yeah. Yeah, it's a violation of statistical thinking, basically, that you're going to... So it's fine if you want to test your hypotheses first, right? This is what I think is really happening. And then you want to explore refinements of that hypothesis in a post hoc kind of way. Actually, that was a great example that Josef gave the other day with the habitat types of the turtles. The first level hypothesis was habitat type affects turtle evolution. And that was okay. But then he found later that actually it is habitat type, but actually only one of them had any real explanatory power. That's totally fine. And that's what you would normally consider as a statistical test before you look at your data, and then a post hoc test of like, oh, maybe that hypothesis could be improved, right? People do that all the time. That's fine. But you don't find the very best fitting thing first and then claim that you did an unbiased statistical test without looking at all the answers first. Okay? So these are great tools, but make sure you use them wisely. So that's one point about the thinking. We have a lot of tools now. Okay, so be careful. Okay, so regardless of that philosophical issue, There are still some pesky lingering problems. So one of them is a little bit of confusion about the root state in the OU model, whether you're using Brownian motion or OU. Okay, and so typically in the early days, we would fit a value for the ancestral state, which is well defined in a Brownian motion. But in the OU, it's a problem, and particularly, so the advantage of fitting it is that results in Brownian motion formally being a special case of OU as alpha goes to 0. The only problem is that the data have little information about the root state, right? It's very hard to predict what the ancestor looked like 100 million years ago when you only have the species today. So these estimates are often inconsistent. So we can fit a parameter for the root state, but it's not consistent because there's no information there. And so as NA found out in 2008, for an OU1 model, the root state and alpha are often correlated. So this makes sense, right? If you're, because you just did these simulations. Okay, so what you have is these phenotypic values, and you have some history, but did it evolve rapidly, or did it evolve slowly for a much longer time? I'm not drawing this well. Okay, let's say that these are phenotypic values. Okay, in present day, and we're trying to figure out, did evolution occur really quickly, or did it start off way back with much lower rates of evolution? You don't have any information about this. You could have had strong selection for a shorter amount of time, or weak selection over a much longer period of time. So that's why you can get all different kinds of sets of parameters that provide equally good fit. And you can get erratic results. And this is especially true when you have a single optimum OU. It's a little better when you have multiple optima because there's a little bit more information, and there's in the phylogeny, you have different amounts of times in different selective regimes, which provides a little bit more information. So so since the root state, this was not a good illustration. The root state could have been here at the mean, or it could have been here and just diverging differently, whatever. So instead of not knowing where the ancestor was, NA proposed, and Aaron actually came to the same conclusion, that let's just assume that the ancestral state is distributed according to the stationary distribution of the OU process. So basically, we're going to just assume that it is among the optima of the OU process. Okay, so where the optimum would be if it was allowed to stabilize over infinite time. So that is very helpful because it gets rid of the problem of estimating an unidentifiable parameter. And Thomas Hansen also came to this conclusion too. So the model fit is better behaved. The slightly pesky problem is that now, formally, Brownian motion is no longer nested within OU. Okay, so when you're doing simulations, you can't just take the OU model written with this assumption and take alpha to 0. It doesn't quite match up. Okay, but it's okay. You just have to understand that if you want a Brownian motion model fit, you have to actually fit a Brownian motion. Okay, so, okay, another pesky problem is that we've discovered that, as you just did this morning, that there's a scaling of the parameters and there's time dependence. So that makes it difficult to compare parameters across studies. In fact, it's kind of nonsensical to compare alphas and sigmas across studies. So what we worked on in this paper that just came out in 2015 are some dimensionless parameter combinations so that we remove the scale dependence of the parameters. So we have dimensionless time, dimensionless trait value, dimensionless optimal values, and basically came up with these combinations of dimensionless parameters that seem to better predict power. And so the first is selection opportunity. So it's alpha times time, depth of the tree. So this is basically the depth of the tree relative to the characteristic time scale of adaptation. So you can kind of think of it as like a phylogenetic half-life. It's inversely proportional to phylogenetic half-life. So it's setting adaptation, scaling it by the time depth of the tree, basically. The next one is this phi parameter, which is discriminability ratio. It's a mouthful. But what it is like, how distinct are the optima in the whole framework of the problem at hand? How well separated are your optima? So if they are well separated, then you will be able to identify them, and you should have more power. Okay, so it's basically, it's the square root of 2 times alpha times the smallest difference between your optima divided by sigma. So the reason why we chose the smallest difference between the selective optima, because the optima that are the closest together will be the hardest to identify. And so that will be the limiting factor between whether you can determine whether it's an OU2 optimum model or a 3 optimum model, if you have some that are really close together and you can't tell the difference between them. Okay, and then the last one is square root of phi. So it's a combination of these two. And basically, it's the signal to noise ratio. So how much signal is there versus noise in your data?
Question.
Yeah. Oh, yes, yes.
So, but if certain opportunity is the AT,
Mm-hmm.
it's a kind of the inverse of any half-life.
Mm-hmm.
Technically, it's not dimensionless, right?
Well, it's proportional to times time. Yeah. Yeah, that's this. Well, alpha is time dependent, so by multiplying it by the time scale, it becomes dimensionless. Yeah.
In the second paragraph,
Mm-hmm.
why do you divide it by sigma?
It's because square root of 2 alpha over sigma describes the standard deviation of the stationary trait distribution.
Sigma is determining how much the optimum is varying.
How much? So yeah, so I should have wrote it like this, but I was lazy, times delta theta. Okay. Okay, so let's see what happens when we try to, so we have to do a simulation study so that we can know what these things really are. And Clay did this massive study. So he generated, and so Aaron didn't trust any birth, death, he didn't trust any simulated phylogenies. So they all look weird. They do. So we took a very large phylogeny. So we took the Anolis phylogeny, which is over 150 taxa or something like that. And then we subsampled and got 40. And so we took these subsamples. We generated 40 different regime paintings of this tree. For each regime painting, we chose 40 subtrees of the specified, of a particular size, so we varied tree size. And then we calculated tree regime and regime metrics. For each subtree, there were 40 phenotypic simulations, so we got 40 data sets for each subtree. And then we calculated this, with a specified, so we simulated these with various selection strength and noise intensity. Then we fit the models to each of these, calculated power, precision, accuracy across the 40 phenotypic data sets. So all in all, there's 14 times 10 to the 6 simulated data sets for each of 6 models. That's why I call it the mother of all simulation studies. And these are the models that he tested. So the true model was OU3, the 3 regime model, and then also Brownian motion, OU1, and then 2 different flavors of a 2 regime model, which was arrived at by collapsing one of the 3 regimes. So these are pretty close, the 2 versus the 3, and then a non-phylogenetic model. So the good news is that the wrong model was very rarely chosen. Oh, and the other thing that really pissed off the reviewers, I don't know why, but we tested the region that's difficult. Okay, so it's already been well established by many people, some of whom are in the room, that it works pretty well when selection is really strong, the methods, okay? So we were interested in the region where selection is weaker or stochastic effects are stronger, so where it's harder, what's the limit of what we can do? And I personally was also very interested in small sample size. So we were looking at 10, 30, and 50 taxa. So, so these are the 6 models, and 0 is bad, and red is selected, blue is not selected. So pretty much only the correct model was selected. Except here, you get a little bit of models selected in the Brownian motion, but note it's when you have no discriminability and very little selection opportunity, which would be very indistinguishable from Brownian motion. So that's okay. Okay, but so that's the good news.
Could you say again what those mean? I kind of forgot. Discriminability ratio.
So this is how distinct the optima are. And this is how strong selection is, yeah.
How strong selection is?
Yeah, in scaled to the time depth of the problem of the tree. Okay. Yeah, so overall, model selection works great. And this is the power. So power, remember, is model selection. How often do you choose the correct model versus other alternatives? And so this is discriminability ratio. How distinct are the optimum? This is how strong is selection. And then this is tree size. So when you have 10 tips, you don't have too much power. When you have 30 tips, suddenly, boom, you've got a pretty decent power. And 50 tips is better. So when you have like your well separated optima and low selection, you still have pretty good power. When you have not very distinct optima, but strong selection, you have pretty good power to a point. When your optima are super close together, nothing will help you, right? These are some of the takeaways, but there's a pretty good region of power. But the bad news is that the parameters are not super identifiable. It's hard to get good estimates of the parameters without huge confidence intervals, is another way to put it. So the best was theta, the optima. This is the best. Everything else was worse. Okay, and in particular, alpha is hard to identify well. Okay, so we have a cautionary tale. The good news is when selection opportunity and strong selection, well-separated optima, when they're large enough, adaptation can be reliably detected. When the discriminability ratio is large enough, power can be high even when selection opportunity is small. So if you have distinct optima, even with weak selection, you can still have decent power. So in this case, tree size and signal-to-noise ratio are good predictors of model power. So in this case, having more taxa will help a little bit. And a strong signal. The bad, some parameters are really hard to estimate accurately. And discriminability ratio, the relative strength of selection in general, discriminability ratio, like how well separated the optima are in general, is better estimated than selection opportunity. In some regions of parameter space, model selection can be quite reliable, even when parameter estimates are way off. The ugly. There is no way of knowing for sure whether you're in the low performance region of parameter space because the parameters are hard to identify. However, it's true that we explored an extremely wide range of parameter space where we really pushed the limits of the model. And so like I keep saying over and over again, most comparative biologists studying adaptation would not be likely to pose hypotheses for data sets in which the optima are difficult to distinguish. Because how would you even know about them in the first place? Okay, so, but this is a simulation study to flesh out the limits of these methods so that you understand, you don't want to bet the bank or bet the farm on the value of alpha for sure. Okay, but so when you interpret your models, really, I think what I would recommend is that you focus on model selection and try not to make, specific conclusions about parameter estimates. Yeah.
You said that data was one of the best parameters estimated.
Data, yeah.
Yeah. Do you think that's because it kind of has a dependency on the phenotypic means, right?
Yes, it definitely does.
Normally you have good power to estimate phenotypic means and not.
I would buy that.
Yeah.
Yeah, I don't really know, but that sounds reasonable to me. Okay, so this is the good, the bad, and the ugly. Okay, so this is, have you seen these kinds of plots? So you can simulate the data under two different generating models. So one is the correct model, one is the incorrect model, and then you take the AICC differences when you generate under, I think this is the OU3 and this is the alternate model. And then you can see where your observed values fall. So basically, if your observed value is far away from the difference with the wrong model, you have high power. If you have big overlap between these high statistical significance. If you have a lot of overlap between these two, there's not much power. Okay, so Clay went further and took this figure and chose parameter combinations. So the star is the true value of the parameters. And then the circle with the confidence bars are what he recovered from simulations of those. So if you're in the good region, you can get a really good estimate of the true parameter values. And you have high power and high significance. Good. If you are in this terrible region, you mostly get back something, well, you have like no confidence in this estimate. It could be anywhere, but okay. But this gives you something like what you would expect, not a lot of significance and low power, a lot of overlap. But the troubling thing is that there are cases, there were not very many, but he explored a lot of the space. And he found some cases where you're in this terrible region of parameter space. But when you reconstruct, when you fit the model, it's going to tell you you're in this pretty good space. And that's a problem. Okay, so this is the misleading part. So it's just something to be aware of.
With the disillusionment
Yeah. Yep.
result, isn't it?
Yeah, yeah. Yeah. So we have these amazing tools, right? But we do have to remember at the end of the day, our data are limited. Okay, so be careful. And also really make sure that you do these resampling methods so that this is the best indicator you have. It's not always going to be truthful to you in all cases, but this is the best we have. Okay.
Can I comment for a moment?
Yeah.
I want to reinforce that because I think we're headed into an era where, with the popularity of OU models, that people are going to take modest numbers of species and try to find out on a tree whether you've got, for example, 12 different regimes on a tree with only 30 species. And I think these kinds of simulations are really important to give you an idea before you do the study whether you're in fact going to have any power to tell apart, for example, different parameter combinations, or in the material that's coming up on multiple OU peaks, whether you really can tell them apart.
Right. Yeah. And so I guess the other thing is, in reality, there may be 12 peaks, but this tells you we can't tell. We can't tell for sure at all. So I totally, wholeheartedly agree with that. And we want to always try to choose, none of our models are full reality, but what is the simplest model that can explain the majority of the variation? What's the most useful model? I think that's, yeah. Yeah.
Would it, I don't know if this is advisable, but would it be any use to test against like a null permutation of values for your particular data set, randomizing the regime weight association, something like that?
I think, well, yeah, you could certainly do that.
It's going to be, that's really useful, but as you say, every study is kind of different and generally underpowered. You're not really going to know what your null distribution is, are you?
Right. I guess I take this is just my interpretation, but I take the paintings as fixed because these are the alternatives I'm interested in. So I'm basically accepting them as truth and seeing, is there any power to distinguish among them? But if your goal was to accurately infer where those transitions occurred, then definitely you would want to randomize that. So I think you have to think about randomizing what it is you're testing. Yeah. Okay. Okay, well, I did want to show you the bivariate stuff, but we only have like 7 minutes, so.
You want to cut it off here?
Um,
Because I want to make a comment. If you're worried
Okay.
about not having anything to go in the 7 minutes. But if you want, if you're trying to decide whether to put in this last section, is that right?
yeah.
How long does it take?
Okay, well, okay, I can do it quick. Okay, so, so after I told you that depressing story, I'm now going to show you a more complicated OU model. Okay, so we have developed a multivariate extension of OU, and it's a multiple optimum multivariate model. And so this is a bivariate case, and so this is vertebrates, and it's testing for an association between basal metabolic rate and genome size. And it's using the data of Waltari and Edwards 2002. Okay, so I'm going to just go through it really quickly because we have 7 minutes. But this is the basic problem. This is biodiversity. We have this huge range of basal metabolic rate and genome size. How do we explain it? Are these values just randomly dispersed? Or is there some sort of relationship? Is the pattern completely driven by the weirdo salamanders? Perhaps also by the unique biology of birds. I mean, they fly. Or is it related to functional constraints resulting from endothermy versus ectothermy? And maybe constraints related to flight. Okay, these are all biologically informed hypotheses. And then do basal metabolic rate and genome size covary? And what the heck does that mean anyway when you get to this level? Okay, so here's my illustrations. 2D, Brownian motion. This is what you get. Okay, Brownian motion. You get a perfect correlation. Okay, anyway. OU with strong selection. Here's the start. You have speciation. You have 2 different optima. And in strong selection, you get pretty rapid divergence. And you can get different clustering of traits. Okay, so with 2 traits evolving, these individual parameters now, of course, become the dreaded matrices. Okay, and so the problem is, I mean, there's nothing wrong with matrices. We just don't have any power. Okay, so anyway, the diagonal terms are selection and drift as usual, but the off-diagonal terms now are very interesting, right? Because they're the correlational effects of trait 1 on trait 2 and trait 2 on trait 1, which of course we're going to assume is symmetrical because that would just be insane. Okay, so big problem is you now have, with multivariate, a superlinear increase in parameters.
And what about sigma? Do you really want a 0, or is that a symmetrical matrix?
It is a symmetrical matrix, but we use sigma squared, so this is sigma.
Oh, and you square it.
Yeah.
Oh, this is like your standard. I see.
Yeah.
Sure.
Yeah, so this is,
Raised to 1/2 power.
yes. This is like a standard deviation of the sigma. Sorry, it's a little confusing. Okay, so we tested these different models. One is the double univariate, which is essentially 2 univariate analyses slapped together. You could just do 2 univariate analyses and get the same output. The second is correlated drift. So you have no correlation in alphas, but you get some in sigma. And then the final is there's correlation allowed everywhere. Okay, so what does it look like? I mean, that was my first question. What the heck is this? So when you have no diagonal terms for sigma and alpha, you get this type of simulation. And when you get correlated drift, you get a slightly different looking final distribution. So you see, the Brownian motion now has become squished. So these things are covarying. So there's no covariance between the two characters. Now there is. Okay, so what does this covariance and drift describe? Well, it's the amount, the eccentricity of the ellipse is what it's describing.
Can I make a comment?
Yeah.
If that's drift to the mean,
Mm-hmm.
then that term, that covariance arises from the G matrix, and the matrix you just drew is proportional to G.
Okay, so correlational selection, what does that look like? So if we add the off-diagonal alpha terms in addition to just regular sigma, we get this, as opposed to having no off-diagonal terms. So I was really excited when we got this, because of course, what does this look like? That looks like, correlated selection, where you can't quite reach your optima because the correlational pull from the other trait is keeping you from getting to where you want to go. That was my interpretation. Okay, so these were our hypotheses. Okay, so this is a hypothesis based on genome size predictions. So salamanders are weird. Salamanders and birds are weird relative to the other vertebrates. Basal metabolic rate, endotherm versus ectotherm, birds, mammals versus ectotherms. And then maybe it's really just the unique biology of each major clade that is describing these patterns. And these are the results. So with the simplest model, we get a huge difference when you exclude salamanders and use the major, what was this? Sorry. Oh, right. So this is major taxa. So the difference between here and here is this is anurans and, amphibians, and this is anurans and salamanders split apart. Okay, so what we learned is separate regimes for each major taxon, splitting salamanders and frogs was the best model. Over 100 AIC units better. It's a really big tree. So when you include correlated drift, the off-diagonal sigmas do improve fit universally. But in this case, the off-diagonal alphas didn't. Okay, so all the adaptive models perform better than Brownian motion. There is a signature of correlated evolution, but it's revealed basically only after accounting for these major differences between vertebrate taxa. And we have strong selection, especially on genome size. Moderate drift. Yeah, after what I just told you, that doesn't, anyway. We have correlated drift, but the off-diagonal alphas did not improve fit. So this is the profile likelihoods for the parameter estimates. So this makes a lot of sense. So we find this type of pattern a lot, especially when it's multivariate. There's just a flat likelihood surface. So this is why it's unidentifiable. You get near the optimum, and then you're just wandering all over the place. Like an infinity is about as good a fit as 20, right? That's why it's unidentifiable. So you need to know what these pictures look like so that you can make reasonable conclusions. So we can say that correlated drift is negative. I'm safe in saying that. we're safe in saying that selection on basal metabolic rate is pretty big. And this is really huge. That's all we can say. We have no idea what it is. So how do we explain the data? There's a stamp of history. The biology of the major groups is tremendously important, shaping their selective regimes over millions of years. There's very strong selection. We don't exactly know how strong, but especially on genome size with moderate drift. And after accounting for these differences, there is a strong signature of correlated evolution. So anyway, that's about all I wanted to go over. This was before we did this massive simulation study.